Welcome to Lecture 11 on the topic of plant growth and development. This lecture will be delivered in two parts, and this is part 1. This lecture is part of the subject Plant Physiology, which is offered as a component of the Bachelor of Agriculture and degree at North Melbourne Institute of Technology. Please visit our website www.nmit.edu.au for information on this subject and the other courses that we offer. My name is Dr. Nikki Cooley. This lecture is Lecture 11, and we will be looking at plant growth and development from a whole plant perspective. This is stage 2 of this subject. In stage 1, we travel through the plant in a virtual way, learning about many of the underpinning concepts that allow plant growth and develop to occur. Please ensure you have completed stage 1 of this subject before continuing on to the stage 2 topics. In part 1, we will introduce plant growth and define plant growth. I have included some video resources to assist you with some of these concepts. In part 1, we will also introduce plant development and differentiate the difference between growth and development. In this slide, you will also be introduced to a plant called Arabidopsis. A picture of its growth from week 4 to week 20 is shown on the slide. This Arabidopsis is used as a model plant, and much research on growth and development, particularly at the gene level, has been conducted on this plant, and concepts from this evidence-based research will be described across part 1 and part 2. Before continuing this lecture, I'd like for you to see an overview of plant growth. The 3 following very short YouTube videos are growth of 3 different plants. One is Arabidopsis thaliana, which is the model plant often used for investigating components of plant growth and development. The second plant is a time-lapse of radish seeds sprouting, and where you can see both the hypocotyl growth and the root growth at the same time. And the third one is the David Attenborough Private Life of Plants growth of fungi. All of these will give you the visualizations, and we will talk in more detail in this lecture about how these components come about. Growth can be defined as an irreversible, permanent increase in size. Here we have an illustration of the stages of growth of a bud. You can see year 1, year 2 and year 3 growth. It is helpful to differentiate between growth and development. Growth is the cell division and cell elongation, while development is the whole process, that is, from germination all the way of the different stages of growth through to death, as illustrated on this image on the slide. An observation that is perhaps quite obvious about plants is that they don't move around. They don't get up and walk, and they don't run, unlike animals. Therefore, unlike animals, they have never evolved anatomy that enables mobility. Instead, plants have evolved a rigid anatomy which is adapted for the capture of light energy and nutrients. Both of these aspects we have explored in previous topics. The rigidity of the anatomy affects how the plant can grow. Cells are added during growth by meristems. In contrast, many animals' cells migrate. The lack of mobility means that plants have to adapt through flexible patterns of development, including the regulated proliferation and differentiation of meristemic cells. Basic plant classification was covered in Practical 1 workshop of this subject. In this practical, we also looked at various parts of the plant anatomy. Please refer to this source when you are learning about this topic, as it may assist in some of the concepts covered here. A saprophyte is a diploid generation of a plant or algae that has a double set of chromosomes. All land plants and some algae have life cycles in which a multicellular haploid gameotype generation alternates with a multicellular diploid generation. In the figure on the slide, you can see a visual representation of a typical saprophyte. This slide draws to your attention the various scales. For example, the whole plant height is 250 millimeters, while the flower height is 3 millimeters. This gives you some concept of scaling and the role that scaling has in plant growth phases. Stages of saprophyte development. The first is embryogenesis. This is the process by which a single cell is transformed into a multicellular organism having a characteristic but basic organization. The second is vegetative development. With germination, the embryo breaks dormancy and by using stored reserves commences a period of vegetative growth. Reproductive development is then followed. Plants make transition to reproductive development. These stages are listed on the slide for your information. In seed plants, embryogenesis transforms a single-celled zygote into a more complete plant embryo in the seed. This process includes many developmental processes: morphogenesis, organogenesis, histiogenesis, which is differentiation, dormancy, and finally germination. Arabidopsis is a small plant used by many plant scientists as what we call a model plant. That is, a lot of time and research funds have been spent on identifying components and physiology of this plant. This is then related to the rest of the plant kingdom. There are 5 stages of Arabidopsis embryogenesis. The first stage is the zygote stage. This is where the fusion of the haploid egg and sperm to form a single-cell zygote occurs. Growth and an asymmetric division give rise to an apical cell and a basal cell. Globular stage is the second stage. The apical cell divides to form an 8-celled globular embryo. Further cell divisions generate the outer layer, the protoderm, which will then become the epidermis. The third stage is called the heart stage. This is where rapid cell division occurs on either side of the future shoot apical meristem from the cotyledon primordia. The fourth stage of embryogenesis in Arabidopsis is called the torpedo stage. Here, cell elongation throughout the embryo and further development of the cotyledons. And the final stage is the mature stage. The embryo and the seed lose water and become metabolically inactive as they enter dormancy. Storage compounds accumulate in these cells. For a visualization of embryogenesis in Arabidopsis, please stop the video here and click on the following link. The URL link to this site will be also available for your convenience on Moodle. The figure on the slide is from the Tays and Zeiger recommended textbook and illustrates the stages of embryogenesis. This visualization may be an easier way for you to remember these stages. The process begins with the initial structures of the apical and basal cells. This is known as the zygote stage and illustrated in figure 1a. Cells continue to divide, B and C, until the production of the protoderm, which is the protective layer. This is followed by rapid cell division producing the cotyledon, as can be seen in figure E. This is then followed by elongation process and is eventually complete with the production of the mature stage. You will note in the mature stage it contains the shoot apex, the cotyledon, the axes, and the root apex. A characteristic feature of seed plants is what is called apical basal polarity. This is a polarity in which tissues and organs are arranged in a particular order from the shoot to the root meristones. Polarity commences in the zygote. It becomes polarized in the intracellular composition. For example, the apical end is densely cytoplasmic, but the basal end contains a large vacuole. The zygote divides to give a short, cytoplasmically dense apical cell and a longer, vacuolated basal cell. The figure on the slide summarizes the processes we have just discussed. This simple visualization may be a good memory aid to remember pattern formation during embryogenesis. It is quite incredible that nearly the entire embryo, thus the mature plant, derives from the apical cell. The apical cell divides to generate the 8-celled globular embryo. The basal cell divides to produce the suspensor, which attaches to the embryo to the vascular system of the parent plant. Only the hypothalamus becomes part of the mature embryo. This forms part of the root apical meristem. In this slide you can see how the apical embryo divides into an 8-cell, then a 16-cell, then an early globular unit. The 8 cells of the globular membrane continue to divide by the periclinical division, that is, parallel to the tissue surface, to form the protoderm. Cells of the protoderm undergo anticlinical division, that is, perpendicular to the tissue surface, to increase the area of a 1-cell thick epidermis. By the early globular stage, broad areas of cells can be related to cell fate: the apical region, the middle region, and the hypothalamus. Now you should have a complete understanding about how the early embryo sac and cell division and cell elongation results into a seedling. The figure on the slide demonstrates the important stages of this process: the development of the embryo sac, the zygote, multiplying from 1 cell to 8 cells to 16 cells, the early globular structure, the late globular structure, transition, late heart, and finally seedling stage. During animal development, substances called morphogens play key roles in providing positional cues to cells during embryogenesis. While with plants, auxin may act as a morphogen in plant embryogenesis. Auxin is a hormone, indole 3-acetic acid, or IAA for short. It can be used to induce the formation of embryos from stomatic cells. Indeed, there is evidence in the literature to support the role of auxin in embryogenesis. The figure on your slide, 16.7 from the recommended text, illustrates the results of when you put various compounds that either inhibit or promote auxin transport, and how this in turn impacts on the developing embryo. In figure 1, an altered morphology was found when culturing in vitro for TENS day in a compound called tranexamic acid, which inhibits auxin transport. Similar failure was found in cotyledon separation caused by distributing the PIN gene, which is involved in auxin transport. Figure B is the wild type, where no change in morphology was seen. The slide illustrates auxin movement through growth. As you can see, the location of the maximum auxin, or the highest concentration, changes depending on what stage the growth is at. Stages shown vary from 2-cell stage, globular embryo, to early heart stage. The meristem is a description that we use often in this subject. It refers to a group of cells that retain the capacity to proliferate, but whose fate is undetermined. There are two major meristems, the root apical meristem and the shoot apical meristem. Both of these meristems have features of defined cluster of cells called initials, and they are distinguished by their slow rate of division and undetermined fate. The image on this slide was obtained from a genetics review paper. It is a scanning electron micrograph of a young maize apex, showing the shoot apical meristem at the top, and in green the youngest leaf primordia, or plastochronin, P1, P2. These are indicated in the green colour in the figure. There is some associated literature I would like you to read with this topic. I'd like you to read from the recommended textbook, Tays and Zeiger Plant Physiology 5th edition, Chapter 3, on the section of the components of water potential that vary with growth conditions and location. This is on page 45. I'd like you to read two sections in Chapter 5. The first is on mineral nutrition excess minerals in the soil limit plant growth on page 79, and the second is on the topic of soil pH affects nutrient availability, soil microbes and root growth, and this is on page 79 also. I'd like you to read Chapter 10, Long Distance Signals May Also Regulate Plant Growth and Development section, which covers pages 218 to 219. And finally, to complete the reading for Part 1, please read Chapter 4 on Gene Expression and Signal Transduction, pages 1 and 2. This is a summary introduction to growth and development and gene control. Please make notes on these readings and insert to your lecture notes here. Once you have listened to this lecture, watched the associated videos and read the recommended literature, you will have an overall fundamental understanding of plant growth. You will be able to differentiate growth and development and understand the differences and how one can measure development. Two important components of plant growth is SEM and REM. Please familiarise yourself with these concepts and what they mean and the important role they play in plant growth. And finally, we've had a look at auxin, the main signal controlling growth. There are other hormones that control growth, and we will be looking at them in due course too. That brings us to the end of this lecture and Part 1.